All right. So we should be good. Got a lot to cover. Exam on Friday. Same format as our first exam. It is just covering the nematodes that we get through. We're not going to get through all of this presentation. We're not going to get all of our life cycles, but that's okay. We do have Oncocerca and we have Filaria in the lab, so we can kind of look through those, and I could ask questions about some of them, but it might be one question, and it would probably be in the lab. So 30, so the first 10 are the multiple answer type questions. The next 20 questions are just single multiple choice questions, just like before. Then we're going to have a section on disease names. So I give you the disease names, you tell me what parasite, and again, I'll give you a word bank of the parasites that we've covered. Then there's the symptoms, the diagnosis aspect, where I describe the symptoms related to the disease, and based on that information, you should be able to have an idea of what parasite is causing that. And then our life cycles. Again, 2 life cycles to draw. You'll have your choice of 2. And just remember that I'm not going to give you a choice of a life cycle where one of them is like really simple and the other one is really complex. All right, so they're going to be about the same complexity. So that way we have approximately the same number of check marks on them. All right? And I won't be here on Friday, so a graduate student's going to administer the exam. She should be up here about 9:50 or so, and she already knows that she'll just, if you want to start at 9:50, you can, and when you're done, you'll hand it in. And same thing, you'll write your name on the answer sheet, and you'll write your name on the test. You'll get them both back. All right, so trachunculus. We did our life cycle, right? So who's the definitive host? Humans. First intermediate host is? Copepods. How do humans get infected?
Raw fish.
No, not raw fish. How do they get infected? Yeah, they accidentally consume the copepod while they're drinking water. All right, so key points for the life cycle. So we said it's going to take about 10 to 14 months to develop this blister, this skin lesion. All right, so when you first get infected, it takes a while. This worm has to develop, and it undergoes that migration. It goes to those, one of those 2 regions, normally the lower regions, before the female undergoes her final migration to form this lesion, this blister. All right. Now, the reason it has to get out is because as this worm is growing, as the uterus is filling up with these J1s, the vulva actually atrophies. So it's no longer functional in these adult females. That means there's no way for these larval stages to get out other than having the cuticle break and having the J1s get released that way. So that's why the body has to actually stick out of the host to deposit the J1s. The vulva is not functional anymore. It's just not there. All right. It does require the copepod intermediate host. We'll talk about that. And we said the female worm is going to die after releasing their J1s because, again, the body wall ruptures. That's how our J1s get out. How do we remove them? Just like this. Just like this. Take a little bit of a worm. Worms are already sticking out. You wrap it around a stick, and then you pull out a few centimeters per day. So, trachunculus. Pathology. Usually this is non-fatal. All right, you have it, you deal with it. All right. Migrating females will stimulate inflammatory response. So you could start to experience a rash, localized rash where the female is. You could exhibit nausea. We didn't fix that. I fixed it on another one. Nausea. Diarrhea, dizziness, localized swelling, so localized edema. all from inflammatory response. This is the migrating female. And then once you get to the surface, you're going to get this painful blister. And this is kind of a good image of that blister. I mean, if you think of a blister, it's a blister. It's a blister. And that blister is forming because of the inflammation, irritation, inflammation of that worm. You get this blister formation, and eventually it starts to itch, and you'll get this intense burning sensation, driving that host to seek out some cool water, which the parasite needs. All right, once that breaks, we're left with an open sore, with a lesion. All right, and that lesion isn't necessarily the problem. I mean, it's going to heal rapidly once the worm gets pulled out, but since it is an open wound, you have the possibility of a secondary bacterial infection. And in most of the places where this occurs, it's pretty likely. It's about 50-50 chance that that individual will get one of these bacterial infections. If left untreated, it's going to be pretty painful. But again, this is something you can treat. Sometimes these female worms fail to reach the surface. They get lost and they don't know where to go. And when this happens, they will just die and our immune system will take it out. But due to the size and the response that's happening, oftentimes you'll get formation of these calcified worms. So, calcium deposits, so you start feeling, a slight bulge or what would feel like a nodule of some sort. Or in some cases, you'll have some abscess formation. So, necrosis happening, leading to some sort of abscess. What's another word for an abscess? Like a pit or something like that. All right. Pathology of this kind of just depends on location when this happens, but arthritis is common. So oftentimes they might go and get lost in a joint, they get calcified, then your host starts to exhibit signs of arthritis, pain in the joints and pain in the movement and so forth. This is the fiery serpent. Probably what that was mentioned in the Bible. It's probably in reference to this worm. Now we've mounted a pretty hard attack on it to try to eliminate this parasite. You guys ready for this? Next one. And this effort was aimed at disrupting the life cycle. So with our complex life cycles, I want you to remember, outside of this class, that in order for the parasite to persist, all of the hosts have to be present. If we can eliminate a host, then we can eliminate the parasite. It can't complete its life cycle. But the other side of that is if we can block transmission of the host, if we know that consumption is what leads to the infection, we can block that consumption and disrupt the life cycle. Parasite can't complete it, and thus we can eliminate it. So for this life cycle, that has been the focus. We know the life cycle requires water. That adult has to get to a water source where the J1s will get into the water, and then these J1s are actually fairly short-lived. They have to get into the copepod. They only have a few days. That's it. All right? So the skin of the host must contact water. Well, you can't tell humans you can't go to water. We're dependent on it. And that's why a lot of the human civilizations in these areas, they were focused around a water source. It's just not possible. You can't tell them, don't go to the water. All right? Water has to contain the correct species of copepod. So one thing you can think about is, well, let's just go kill off all of the invertebrates. But if you do that, you're going to do a host of other things because those copepods are serving as a food item for larger predators. And those larger predators are likely being a food source for the humans. So one, it's kind of an ethical question, should we really do it? But number two, those that have tried have failed. They failed, all right? So it's hard to go out and just eliminate a single species without causing this ecosystem crash. So that's not correct, but we do know that humans must accidentally consume a copepod, and the most likely situation when this occurs is when they go to that water source to drink. So the eradication efforts have focused on educating people about the disease and providing or showing them how to filter their water. It started out as being something where you tell them, instead of drinking immediately out of that water source, out of that lake, take a bucket of water and then pour it through into a funnel where you have a t-shirt. That t-shirt's going to capture most, if not all, of the copepods. And then the water that you collect after you filtered it through the t-shirt is going to be great. Nylon tights are the other thing. Sounds great, but hey, in these areas, people are not wearing nylon tights. All right? So what has come up with are these life straws. This was a great thing, because if you're out in the field, you need the drink of water, you're not going to get a bucket. You're not going to carry a bucket around so you can filter it. But if you attach these things to strings, now you can get people to carry it around their neck, and when they go to the water, they can easily drink it. It's just a simple filter. It's blocking the copepod. And it's been really successful. Those numbers that I gave you, we've really almost eliminated it. Came close. They announced, hey, we successfully eliminated dracunculus, but it popped up. Why? They started saying the dogs were starting to serve as a reservoir host. Can't get a dog to drink through a straw. If you can, take a picture, post it on TikTok. I bet you'll go viral. All right. But interesting thing is that these worms are genetically identical to Dracunculus medinensis, which is the human parasite, which is arguing that this is not a different species. This is the same species. It's now going into dogs. Is this a host switch? Maybe. Or maybe it's just it didn't need to go into dogs, or we never noticed that it went into dogs. All right. This isn't terribly gruesome, but yeah. All right, moving on. So we're going to switch to a superfamily, Filaroidea. These are collectively called the Filarial Worms. It's going to include the families Filariidae and Oncocercidae. The parasites in this group are going to use arthropods as intermediate hosts. Whoa, hold on here. Aren't crustaceans arthropods? Yeah. Crustaceans are part of the arthropoda, right? Yeah. These are our insects. That's what we're getting into. All right, so most of these parasites are going to deposit J3s onto the skin of the host, and then you're going to have some insect that comes around and ends up picking it up somehow. Other species are going to utilize these Microfilaria, and the Microfilaria are going to be ingested by our insect host. So what's a Microfilaria? Microfilaria is a juvenile stage that is not as differentiated as a J1. It's sometimes considered an advanced embryo. All right, so we're not quite at that J1 stage. Right, they don't have the characteristics of a J1. And we had these Microfilaria. This was Microfilaria of Thyrofilaria immitis, so Microfilaria of dog heartworm, parasite that causes dog heartworm, that you can check out. And you'll see that there's not a whole lot there. Usually when you look at them, you can identify cells, nuclei present in there, but you don't see those in the J1s. J1s are advanced. They look like nematodes. All right, so members of the filarial worms infect all classes of vertebrates except fish. Fish don't serve as hosts. Why is that? Why is that?
There are a lot of bugs in the water.
Have you collected bugs in water?
Yes.
All right, so be more specific. Be more specific on bugs. What bugs are you talking about? Like blood-feeding bugs? Probably. Yeah, you don't see a whole lot of those. So yeah, they're not in fish. Will they get to fish? Who knows? Maybe in a couple million years, they'll evolve. So some of these species, some of these filarial worms rely on symbiotic bacteria. All right, key one is Wolbachia or Welbachia. I'm not sure what. Who's in that zoonotic diseases class? Is anyone here? No, because it's at the same time, isn't it? I think it's at the same time. Bummer. What about pathogenic micro? Did you guys talk about Wolbachia? In general micro? All right, yeah, so this is a rickettsial bacteria. It's similar to what we call your rickets. Right? But this is a symbiotic bacteria that we see in some of these nematodes. And it's not just, it's not just going to be in filarial worms. It could occur in some of the other worms. And that's how its discovery was somewhat accidental. Worms had pinworm infection. Or mice in a lab had pinworm infection, mouse colony. They also were exposing them to, treating them with antibiotics. So they didn't realize it at the time, but the antibiotics killed off the Wolbachia. And without the Wolbachia, those pinworms couldn't survive. So there was this symbiotic relationship. How they get there is transovarian transmission. So the Wolbachia from the female worm is going to transmit them to her offspring. They don't pick them up from the environment. They get transmitted when they're born. Kind of an interesting part. All right, if they lose their symbiont, yeah, reproduction is going to be impaired. So it's important to know them. Ready? All right, so our family that we're going to talk about is the family Oncocercidae. These will live in the tissues of amphibians, reptiles, birds, and mammals. No fish. Most of them have no medical or economic importance to us. So why do we care? Well, a few of them will cause pretty gruesome and debilitating diseases in humans. The two that we have are Wuchereria bancrofti and Oncocerca volvulus. All right, so we have Oncocerca and Filaria in the lab. So these two can cause pretty significant disease in humans. Filaria is a pretty significant parasite in dogs. That's why we give them heartworm meds every month, or you should give heartworm meds every month. All right. Ready? We're going to start with Wuchereria, I believe. Yeah. So I just have the diagram, the juvenile, the microfilaria of Wuchereria. It's got a cuticular body sheet. Style it. Not as developed as a J1. All right, we ready now? All right, Wuchereria bancrofti. This is a causative agent of elephantiasis. You've probably heard of elephantiasis. Have you? No. Talk about pathology. So humans are the only known definitive host of Wuchereria. There are no known reservoir hosts. This parasite is distributed across the equatorial belt. It used to be just the equatorial belt in Asia, let's say South Asia, but it was brought to the Americas during the slave trade. It was introduced into Charleston, South Carolina, where that was a hot spot of infection. Until about 1920. Can't even say the colonists. We're past that time. We're, past the Revolutionary War, past the Civil War. But at those time periods, this was a hot spot of infection. Charleston, South Carolina had Wuchereria. You had individuals exhibiting elephantiasis. All right? But then in 1920, it vanished, it disappeared. Probably related to the mosquitoes. Something probably happened, maybe a cold winter or something like that. That's kind of a guess. So I do have a diagram of the mosquito because we can lay out parts. It'll become useful. So mosquitoes bite. They've got your palps, your antenna, and you have the palps, and you've got your proboscis. The proboscis is what penetrates in to pull the blood, and it's covered by a sheath. All right, and then you'll have your labial palpus up near the head area. Right. As part of this mosquito, they do have salivary glands, and you have intestines, midgut, hindguts. All right, so I've got the diagram there to kind of help you place parts of our life cycle. So let's go through this life cycle. And this one is fair game for the exam. Wuchereria and Filaria is not. All right. Where are we going to start? Guess we're going to start over here. We're going to start with the Microfilaria. All right, Microfilaria, they're going to be in the peripheral blood. Right, they're in the circulatory system. They're going to be near the surfaces, peripheral blood. Mosquito's going to come around, take a bite, and ingest a Microfilaria with its blood meal. All right, so we're going to have Microfilaria in the gut of this host. This Microfilaria, this is our mosquito. This will be our human. All right, so this Microfilaria is going to get out of the gut. It's going to migrate. All right, where does it migrate? It migrates to the thoracic muscles. All right, so it's going to migrate, and when it gets to the thoracic muscle, it's going to transform to a J1, molt to the J2, molt to the J3 stage. All right, so let's talk about this. They were in the gut. They went to the muscles. The thoracic muscles are, partially controlling your flight. They get in here, and it takes about 8 days of development to get to the J2 stage. At the J2 stage, these things are called sausage worms. Why do you think that's the case? Because what?
Are they linked together?
Good guess. We didn't get. They're not going to be linked, but they look like sausages. So our worms are normally long and thin. These are going to be short and stout. All right. Short, stout, sausage shaped. All right. But these things have an anal plug. The anus is blocked up. What does that mean for the worm? Can't defecate. And you'd say, well, if it can't defecate, then how's it going to eat? Well, it's a good question. It still feeds. But the anal plug is one of those keys that say, okay, we are at the J2 stage at this point because we have this anal plug. It feeds, gets to this J2 stage, and then it's only at this stage for 2 to 4 more days before it undergoes the molt to get to a J3 stage. In which case we are now slender and filariform. So slender and more thread-like. All right, we get to our J3 stage, and then we're going to migrate. So remember, we're getting ready to get to our new host. Where does it migrate? The proboscis sheath. It's a sheath that covers the proboscis. It's also called the labium. For you entomology students. Right now, so this is going to take about 10 to 12 days post-infection is when our mosquito is now infected. So our host is going to feed on a human. Our J3s are at the proboscis sheath. They will come out and they will be able to enter the wound caused by the mosquito. All right? So we have our J3s going to be in that wound and try to get to the blood. It then utilizes the blood to get to the lymphatic system. And it gets to the lymph node, a lymph node, where it finds its way to the afferent lymph chain. What is the afferent lymph chain? So that's our lymph node. We're going to have channels going in and channels going out. Which one's the afferent? The what?
The one going in.
Yeah, the one going in. All right. So it's going to get to that afferent lymph channel. It now is at its spot where it's going to molt to the J4 and then molt to our adult. Afferent lymph channel. There's that one going into the node. All right. It takes about 6 to 12 months for our adult to get here and mature and start releasing Microfilaria. Now, the Microfilaria that get released are in the lymphatic system. They need to get to the circulatory system. All right, so that means they're going to have to get out of that lymphatic system. They're going to have to burrow out and into the blood vessels. Remember that. That could be a cause of pathology. Now, what do we see with this life cycle? Like an adaptation. Adaptation for transmission is periodicity. Periodicity, I should put that into here in our blood. We see distinct peaks and declines of microfilaria in the blood through the day. The peak is between 10 p.m. and 2 a.m. Why is that?
Mosquitoes are most active.
mosquitoes are most active. Do you get bit in the middle of the day? I know I get bit. But you're close. What'd you say? Remember that. What are we doing? What are we doing at this time? Yeah, so this is the time where we are generally sleeping, which when you're sleeping, mosquitoes that can go all out. So this seems to be coinciding with the time when we are relaxed. Now, what's the trigger? Good question. It's a good question. We don't quite know. It could be oxygen tension, could be body temperature. So our body temperatures tend to fall when we're sleeping. Oxygen tension tends to go down when we're sleeping. Those could be the triggers. We just don't know what's causing this periodicity, but we do see it. So if we think we have Wuchereria, the best time to try to draw our blood to look at blood smear is to try to collect it sometime at night. Questions? No? All right. Not terrible. All right. So females live in the afferent lymph channels. Another adaptation for transmission is how long they live. These adults are going to live and shed microfilaria for 5 to 10 years. It's a long time. It's kind of like schistosome. And the reason's probably because successful transmission is low. So what they try to do is shed the microfilaria for a long time in the hope that at least some of them will get to a mosquito. Ready? All right, pathology. There are several names for this disease. Lymphatic Filariasis is kind of a generic name because it's infection of the lymphatic system by filarial worms. There's a couple other ones that could also infect the lymphatic system. About 90% of lymphatic filariasis is caused by Wuchereria bancrofti. So they've called that, instead of just lymphatic filariasis, they call it bancroftian filariasis, which is lymphatic filariasis caused by Wuchereria bancrofti. All right, that's your disease name. Once you start getting some of the symptoms show elephantiasis, which is the buildup of lymphatic fluid leading to the thickening of the skin. Scroll ahead because I don't got some. Do have a picture. All right. So elephantiasis is the buildup of lymphatic fluid leading to the thickening of the skin. Why do you think we have this buildup? So female worms are in that afferent channel. That channel goes in to the lymph nodes. And it makes sense to say that the females are blocking it. That's not really the cause. It's going to be caused by the juveniles, by the microfilaria. They have to get out, and they're going to be doing some damage. And once they do the damage, you're going to have thickening of the lymphatic system, thickening of these channels leading to blockages. So that's going to cause lymphatic fluid kind of build up. Like, we should be draining our body of this excess liquid. It can't drain, so it's just going to collect in the tissue, and we're going to see this gradual enlargement. That stresses our skin. Our body responds by increasing fibrinogen, thickening of the skin. All right, and I do note that just because it's elephantiasis doesn't mean it's Wuchereria bancrofti. Some other things will also cause the symptom, what we would call elephantiasis. All right. Now, I've often presented our pathology as a progression. right? With our infections, think about all the other nematodes. You've got this migration phase, then you've got your GI tract phase, and so forth. This disease, Wuchereria, could cause this progression of symptoms, asymptomatic inflammatory phase, and then an obstructive phase, but it doesn't always have to do this. We might not see some of these. We might not know we have anything until the obstructive phase. So let's talk about the phases. So the asymptomatic phase, individuals in this case have high levels of microfilaria in their blood. Microfilaria would be the term. And what we see with this phase is that Th1 inflammatory responses are suppressed and Th2 responses are stimulated. So the guess is that the microfilaria are probably going to be successfully eliminated with the Th1, but that gets suppressed. We suppress it, or the worm suppresses it. And this is going to exist for several years before we start to see inflammatory reactions increase. This inflammatory response could be linked to worm antigen. Or, I'm sorry, we'll talk about that. We'll talk about the inflammation, the antigens and stuff. No, but this asymptomatic phase, not everyone's going to get it. And usually, the people that are most likely to have this asymptomatic phase are going to be those individuals that were born and grew up, or lived, and their parents lived, in areas where the parasite normally transmits. So the parents, the mother in particular, is exposed to Wuchereria, maybe she had it, and thus some of the worm antigen gets to the fetus, and then the newborn doesn't really recognize the microfilaria as normal. So when the individual is born and they do get Wuchereria, it's like, hey, I don't recognize it, I'm not going to respond to it, so we are asymptomatic. We have a name for those individuals. They're called endemic normals. So they have no symptoms. Some of them, you can't even find microfilaria in there. But we know that they're infected because you can detect the antigens. Not only that, but some of these individuals could develop inflammatory reactions later on in life and obstructive symptoms. So they could develop that elephantiasis, even though they really didn't exhibit any of the asymptomatic signs or the inflammatory signs. All of a sudden, they just start developing this obstructive phase. And it's thought could be due to this. Developing fetus was exposed to the antigen. Visitors to the area, less likely to encounter this. We haven't been exposed. We go there. We're going to probably exhibit this inflammatory stage. We wouldn't have this asymptomatic phase. Our body would be fighting. All right. So if we go over there and get it, I'll say Vietnam. Go to Vietnam because we've had several of our soldiers that were in Vietnam come back with this. If we do get it, we'll probably exhibit this inflammatory phase, the acute phase. All right, so initially, probably wouldn't exhibit anything until the adult female starts to develop and starts to release microfilaria. The reason we get the inflammation is because of the antigens attributed to the adult worms. So the female's releasing some waste or whatever, all right? We'll also have some inflammation due to the invasion by bacteria on the skin surface, but it's also thought that the Welbachia that lives inside of this worm is also generating some of this inflammatory response too. So it doesn't seem to be only the worm or only the bacteria, it's probably both that are leading to this inflammatory response. The inflammatory response is in response not only to these antigens, but will also start to get some damage as the microfilaria burrow out of the lymphatic system and into the circulatory system. When you have that damage to the vessels, the body's going to try to repair itself, and it calls in, the immune cells to help do that. And what we could develop is something called lymphedema. This is obstruction of lymphatic ducts leading to a buildup of lymphatic system. This is our filariasis, lymphatic filariasis. Right? It's associated with periodic inflammation of the lymph nodes, suggesting that we're getting, some sort of damage, something that's calling in effector cells to that area. And we're going to exhibit these chills and fevers. You're going to exhibit swollen, tender lymph nodes. Maybe tenderness along some of the superficial lymphatic vessels, those that are closer to the skin. In some cases, you'll have very painful lymph nodes. Just kind of depends on where they're at, which lymph nodes are getting hit. All right, and you're going to have then with this obstruction enlargement of a body part. Here it's the leg, the lower leg. Lower legs are pretty common. This is indicating that you've got a blockage. Your body is not draining the excessive lymphatic fluid. In males, you can have a condition called hydrocele, which is lymphatic fluid ends up getting forced into the tunica vaginalis of the testis or spermatocord, which means you've got an enlargement of basically the scrotum. If you do a search for elephantiasis, I'll warn you, you'll probably see some of those pictures. But I won't show them here. See, YouTube has trained me well. All right, and this is a diagram that kind of shows that. So normal lymphatic system, you've got your different nodes, you got your tracers, they travel. Here's a case where it can't. Tracer gets stuck in a lymph node or in lymphatic vessels. It can't drain properly. So this lymphatic fluid in our tissues can't get out. It's actually a pretty interesting paper. All right, so we already talked about the bacteria found on the skin. Sometimes they get into the lymphatic system. They can cause inflammation. All right, and all of this information is related to microfilaria along with some of the bacteria. And I've got a few more things. Symptoms could be orchitis, so inflammation of the testes, usually with sudden enlargement and considerable pain. Epidermitis, inflammation of spermatocord, hydrocele, that was on the last slide definition. And then also proliferation of cells lining the lymphatic system. Repair mechanisms, you just cause more cells to grow, again, leading to additional blockage. When you have these inflammatory episodes, microfilaria could disappear, so you no longer find them. And it's thought that you're getting this blockage around the worm, and it's going to be harder for the microfilaria to get out and enter the blood. So this is all part of that acute phase. And I'm just presenting these terms. I'm not going to ask these terms, but the description of what some of the inflammatory symptoms may be, that could be useful. Ready? All right, the obstructive phase. It is associated with blockage of the lymphatic system due to the cellular proliferation of the vessels and then also infiltration of immune cells. You can have lymph varices. These are basically, you've probably heard of varicose veins. These prominent veins that are enlarged and visible through the skin. Well, these are lymphatic ducts. You can see them. All right, and it's due to lymphatic fluid backing up in those ducts and stretching them out. Chyluria, that's lymph fluid in the urine. All right, and you notice this because the urine will be milky colored. It has some lymph and emulsified fats in it. Oftentimes you'll see some blood in that urine as well. And it's because when you have blockage of these lymphatic ducts, you can have some of the lymphatic fluid back up into the kidneys. And that's why they end up in the bladder and so forth. And then you can get this elephantiasis, which basically transforms the organ. So you've got continued backage of the lymphatic vessels, all right, lymph fluid buildup, and then you're going to have the thickening of the skin and stuff and the tissues. So basically, the organs start to become mostly connective tissues, granulomatous tissue and fat. Skin's going to be thick and cracked. Most likely places, so in men, it's often in the scrotum. It'll be in the legs and can be in the arms. In the women, it's primarily legs and arms, but occasionally it'll happen in the vulva and the breasts. I'm not too sure why you would have these differences in the sexes. Wuchereria bancrofti. All right. I'm going to fly through these next ones. We probably won't go over the life cycles. But for the lab, we've got at least the lab manual has write-ups on both of these. So Onchocerca volvulus, it's in the equatorial belt, so Africa, South America, and Central America. It's thought that it was introduced into the Americas via the slave trade. So historically, it was in Africa. This is the causative agent of the non-fatal disease onchocerciasis or river blindness. So I've been told that in some of these villages where Onchocerca volvulus lives, you'll see these older individuals being led around by young kids because they're blind. And it's the parasite was doing that. All right, so the life cycle, they're going to live in subcutaneous connective tissue. That's their onchocercoma. All right, it's in their subcutaneous connective tissue. The microfilaria will be in the tissue fluid. You're going to have a black fly bite the host and ingest it. Get those microfilaria, and then you're going to have this development. Again, they're going to go to the thoracic muscles, get to the J3 stage. You also have the sausage worm stage as well. All right, and then they're going to go to the proboscis sheath. So just like Wuchereria, and then when that black biting fly attacks, you're going to have delivery into the body, and they're going to basically stay in these subcutaneous connective tissues. They're not going to undergo this complex migration. And once you get there, they're going to mate. You're going to have the female start to develop, start to enlarge, and it forms this onchocercoma, the subcutaneous nodule where the worms are located. And it's mainly composed of collagen fibers. So we had that word list that said, host-derived encapsulation, I believe. The collagen fibers, that entire thing is a nodule, and it took a slice. All of the area around the worm is collagen. All right. So these are going to exist as pairs. Maybe they'll exist as groups. So I said normally it's like one worm. It's an easy way to think of it, that the worm is wrapped up, but that could have been 2 or 3 worms all wrapped up like in a plate of spaghetti. Where we see these onchocercomas develop varies by region. So in Africa and Venezuela, most of these things develop below the waist, so in the legs. But in Central and South America, most of them happen above the waist, so in the head and neck region. And just like Wuchereria, humans are the only known definitive host. Kind of interesting. This suggests there may be like 2, maybe subtypes. I don't want to call them subspecies, but 2 genetically different populations. Pathology. Again, you have the onchocercoma that's caused by the adults, and you'll have this pronounced. It becomes more pronounced when it's close to a bone because it's going to stick out like that bruise. But I would call it a bruise. This nodule becomes much more visible. But again, it's little harm. The worm's in there, but you're not going to see a whole lot. But because you're getting this increase in collagen, skin is going to lose the elasticity. So when that happens, you can get something called hanging groin, which is a sagging of the groin into pendulous sacs, often containing the lymph node. So if you can imagine, onchosarcoma is going to be down in maybe at the waist. You've had increase in collagen fibers. You've lost elasticity. Any fluid that gets in there is going to gradually thicken or gradually stretch the skin. What sets this apart, from Wuchereria is that we don't see inflammation of the testes at all with this, even though you have microfilaria and potential for damage to the lymphatic system. Microfilaria do cause some issues. The dead microfilaria, they're also going to release their Welbachia symbionts. That's normally the main cause of pathology, and that produces something called Oncodermatitis. All right, so the severe dermatitis caused by Onchocerca microfilaria. It's intense itching, could lead to secondary bacterial infection, scratching and stuff, and you could get this abnormal pigmentation. So this is a normal leg. This would be this oncodermatitis. And this leg is also exhibiting some lichenification, thickening, discoloration, cracking of the skin. So again, increase of your collagen. Definitely microfilaria can go to the eyes, causing blindness, hence its name, river blindness. Filaria is the heartworm. You can look it up. Key thing, highest prevalence is correlated with mosquito densities. So more mosquitoes, higher prevalence of heartworm. And heartworm can only transmit when temperatures are above 57 degrees because mosquitoes need that temperature to start developing. So in winter months, if you go up north, if you're in, let's say, Wisconsin and Minnesota, vet tells you you need to take heartworm meds every month. Too cold up there. Too cold over the winter. But they tell you to do that because you'll just get into a pattern and you won't miss it. You won't miss it.
Will this be posted by this evening?
Which one?
Today's lecture?
Yeah, it should go live. But yeah, I'm not going to ask you any of the life cycles. I'm presenting these as, for the lab, some of the stuff in the lab. But what we're going to do is pick up with the Acanthocephala and move forward. Keep moving forward.
The quizzes go live today, right?
Yeah, they should be live. What I think I'll do is maybe post. I haven't separated the life cycles. I had a quiz for all of these life cycles. I don't know if I can pull out only Wuchereria and Dracunculus
Make it practice.
and do a practice. Yeah, I'd have to do it with the other quiz as well. Just pull out those. Only cover up through Wuchereria, because Onchocerca and Filaria, that'll just be lab questions.
Can you go back 5 more slides?
Which one?
5 more slides.
Hold on. Backward.
Yeah, there. No, there.
This one.
Yeah. Thank you.
All right.